Hello, everyone. Today's lecture will be on cell development. Okay, so again, here's my information along with my email. If you ever want to get in contact with me, please feel free. I also recommend you join our group me. It's an easy way to get a hold of me or any of the other tutors, as well as many of the other students. If you have any questions. Okay, so learning objectives for today. First, we're going to be talking about how do cells specialize. So you have many different types of cells in your body. You have blood cells, liver cells, bone marrow cells, neurons, all these different types of cells. But it didn't always start out that way. You started off from just a zygote, and eventually you worked your way up to all of these types of cells. So how did that happen? Next, we're going to talk about cell migration. Again, once all of these cells specialize, they move to exactly which organ they need to move, right? So how did that happen? Next, we're going to be talking about what is apoptosis and why is it important. We're also going to talk about how does the environment affect gene expression during development. Okay. Moving on to the content. First, about cell specialization. So similar to people's cells mature and get jobs as they grow older. Unspecialized and what we consider immature cells are called stem cells, which you've probably heard before. They have the potential to specialize or differentiate, which is the biological term, into various cell types which carry out specific functions in our body. There are embryonic stem cells, which are only found in embryos, obviously, and adult stem cells, which are in adults, right? And these two different types of stem cells have different capacities and capabilities, specifically for differentiation. There are three capabilities or capacities stem cells can have. Firstly is totipotent stem cells. They can differentiate into any cell type, including extraembryonic and placental cells. These are only really found in early zygotes, right before you even form an embryo, because these can form the placenta and the extraembryonic cells. They're not found inside the embryo, so they could be also found outside the embryo. So this is found only in really zygotes in the very beginning of development of humans. Next, pluripotent stem cells can differentiate into any cell type found inside our bodies. This excludes all of these extraembryonic, placental cells and all of those, but it can turn into any other cell type that you can find in your body. Again, like blood cells, brain cells, liver, kidney, whatever, name it. These are found mainly in embryos prior to birth. However, with technology, it is possible to turn differentiated adult stem cells into iPSC or induced pluripotent stem cells. So you can actually turn an adult cell or like a specialized cell, like a liver cell, back into a pluripotent stem cell using technology. And that's what the I, induced, stands for, right? And the opposite of that, the natural version of that, is called embryonic pluripotent stem cells, which are obviously found in the embryo stage. Next is multipotent stem cells. They can become more than one cell type, but are much more limited than pluripotent stem cells. So they can't differentiate into anything, but they can differentiate into multiple things based on where they are and what they're programmed to do. So these are what most adult stem cells are. So talking about adult stem cells, they're much more limited in their capacities than embryonic or induced pluripotent stem cells. They're only really used to replenish cells, which are made very frequently. Most common example are blood cells made in the bone marrow. So the bone marrow has a lot of adult stem cells, and I'll show you in the next slide a picture of everything that these stem cells can differentiate into, all of the different progenitors, but it's basically all of the different blood cells that you have in your body. You have many, as you know, you have many different components of blood, and there are many different blood cell types. Other examples can include skin, brain, reproductive organs. These make also a lot of, they replenish a lot, so they also have a lot of stem cells, as well as other organs as well, like your colon and other organs also have stem cells buried deep within that replenish the cell layers. Okay, so the multipotent stem cell in the bone marrow can differentiate into many different blood cell types, including macrophages, erythrocytes, which are red blood cells, granulocytes like neutrophils, basophils, eosinophils, T cells, B cells, natural killer cells. Those are your immune system cells. Okay, you can see a picture right here. So the multipotent stem cell is right here. Oh, sorry, it's black. Here, I'll make it red. Right here, and it can differentiate into these two types, right? The primitive progenitor cells, and again, these can also differentiate into different things, right? As you can see here, but it goes to this committed precursor cell type, and this is where it kind of is like it's committed to going down this one line of specialization. So, for example, if you want to think of it in humans, this is like the kid. These are like the kid, and he grows into a teenager. He can turn into this type of teenager, this type of teenager, depending on what he's raised in, what environment he's raised in, like what these factors are. You can see use these factors, IL-7. I'm sorry, it's clear. So these different factors, based on these, he can determine what kind of teenager he turns into. And this committed precursors, like if he goes to college and he decides to major in like pre-med or biology or something, he's committed down this line now. So he's sticking to this line. He's still not, for example, he's still not a doctor yet, but he's committed going down this line. And then this again is caused by different factors and different various factors in life. Okay. And then you continue down. And you get to lineage committed cells, and this is like, for example, again, he decided that, let's say he's in med school now, so he's committed to this specific career path, and he's committed to this specific pathway for sure. He's just not exactly there yet. So, but he is like one step away, and that would be going to these megakaryotes, karyocytes, which get broken down into platelets, or these erythrocyte progenitors go to erythrocytes, red blood cells, like I mentioned. This one, monocytes, go into obviously the monocytes like macrophages, dendritic cells. Granulocytes goes into basophils, neutrophils, or eosinophils. This one goes into T cells, natural killer cells, B cells. Okay. And again, these are just different factors. All of these are different factors that would cause it to go down that pathway. Okay, and you don't have to memorize this chart. May be useful to know it like very generally, not actually just for stem cells, but for blood. And you should know what most of these cell types do. You should know what like T cells, natural killer cells, and B cells do, how they're related to the immune system. Obviously, you should know what red blood cells are, platelets are, loosely. These granulocytes and monocytes also very loosely, nothing specific, I think. For them in terms of like blood, their role in the blood. Okay, so what is the mechanism of this specialization? So during development, cells receive orders to differentiate due to various differentiation factors, which tell them what to turn into. Again, like I mentioned, like the chemicals and stuff. There could be other factors. There could be environmental factors. There could be chemicals, biochemical signals. These are not fully researched yet, and the exact details are way beyond the scope of the MCAT, so you don't have to know them, but just know they, based on differentiation factors, they turn into different things as ordered, essentially. When these stem cells reproduce, they undergo something called asymmetric cell division, which is kind of important. It's a form of mitosis, but the product of asymmetric cell division is two daughter cells which are not identical. This is because one daughter cell remains in like the stem cell like has stem cell like features, so it can continue making new cells. So it's essentially like the spawning point, right? Whereas the other one will be the appropriate cell type. So whatever cell type you're making, like a liver cell or something like that, and it'll lose its stem cell properties, so it won't be able to reproduce or specialize anymore. But it'll be able to it'll gain all of the functions of the cell type that it's meant to be, like the liver cell function. Okay, so it makes, here I can draw a picture just to illustrate. So you have a red stem cell. It undergoes asymmetric cell division, and you end up having, I'll make this blue actually, and you end up having two children. One is, so I'll draw it like this. One is the liver cell, and it has all the functions that a liver cell would have. right? But it doesn't have the functions that stem cells have. It loses that function, and another would continue to be a stem cell, right? And this one would, again, split more, and then so on and so forth, like that. Essentially, it'll continue going that way, right? And that's how you make more liver cells while also kind of keeping your spawn point set to spawn more of those cell types, essentially. Okay. In adult stem cells, this happens via signaling cascades and cytoplasmic factors, which allow for this asymmetric division. You don't really have to know the specifics and the details. Those are, you might have learned that in your cell biology class if you took one in undergrad, but it's not really that important for the MCAT. Just know that there are signaling cascades and cytoplasmic factors which tell one to remain the way it is, another one to change. In adult stem cells, this process is slightly different. It's regulated via epigenetic mechanisms, which control gene expression in the stem cell differentiation. Again, remember epigenetic is so all of genes in every cell, your liver, your brain, your heart, your blood, everything has the same DNA. The reason it performs different functions is through gene expression. You only express liver-based functions in liver cells, and like brain functions in your brain, obviously, without expressing your liver functions. So the way it does that is through epigenetic. So things attach onto the DNA or control the DNA somehow, so that it doesn't get affected. So essentially really latches onto the DNA, or it alters like the shape of the DNA in some way, like condenses it, uncondenses it. That's your methylation, acetylation, those types of things. Control which genes get expressed and not. And that's how you control stem cell differentiation via epigenetic factors in adult stem cells. Okay, next let's talk about cell migration. So during the embryonic stage prior to differentiation of pluripotent stem cells, the cells migrate in response to chemical signals to form the three primary cell layers, the ectoderm, mesoderm, and endoderm. And I put here what they are: the outer layer, middle layer, and the inner layer. Once these cells in these different layers differentiate into appropriate organ cells, they also migrate to form the organ morphology. So it continues with migration. And so, for example, the ectoderm would be things like your skin and those like outer organs. Mostly the mesoderm would be your kind of like middle organs, and the endoderm would be like inner organs. And you can look up, if you would like, you can look up a list of ectoderm, which organs come from the ectoderm, which from the meso, which from the endo. I don't think it's very important, but if you want to, in case just for your own knowledge, please feel free to look that up. And again, once they differentiate into their appropriate organ cells, they migrate to form the organ morphology. So, for example, how does your hand form like these like finger things, right? Well, your ectoderm has to make like skin cells and bone cells, then it has to migrate to this area, and then it like forms these specific shapes, right? And then it gets rid of any other shape, like any like literally like cuts it out like out of like marble, like chisel. You can chisel it out of marble. Essentially, that happens. It just removes everything that's not in the right shape. Same thing happens with like your liver. It forms. Yeah, you get the idea. Okay. Next, let's talk about cell death and regeneration. So we talked about development and how cells are made. Now let's talk about how cells die and regenerate. So all cells have a regulated and programmed suicide mechanism called apoptosis. This is only found in multicellular organisms, as it sacrifices the viability of the specific cell for the sake of the whole organism. Obviously, the first natural instinct any living thing has, whether it be a human, animal, cell, anything, bacteria, virus, anything, is it wants to live and it wants to reproduce. Those are pretty much the two main things. In order to form a multicellular organism, the cell has to give up both of these basic instincts. It has to go beyond both those instincts. It can't just reproduce as much as it wants to, because that would harm the organism as a whole. That would lead to cancer. And so a lot of those mechanisms are controlled, and the cells in your body basically sacrifice that instinct to reproduce in order for you as an organism to survive and for you to reproduce as well. The second is to live. It also gives up that because it knows that if it suffers damage or if something happens to it that might harm the organism, if it gets out of control, it will undergo apoptosis, which will be a suicide mechanism, and it'll give up its life. And obviously that goes against the instinct, but it does that again so you don't, the organism itself doesn't get harmed as a whole. This is usually done in situations of extreme external stress, or in the case of extreme DNA damage. Essentially something that can't be repaired. Like I mentioned, it has the body and the cells, they have DNA repair mechanisms. They have like mechanisms if, for example, like the outer membrane gets damaged or something like that, like external mechanism for stress. But sometimes it gets to the point where the built-in mechanisms can't fix it, and if it continues, it'll do damage to the whole organism. For example, if you undergo necrosis, which is unnatural cell death. So, for example, if you like burn your hand or you like get hit really hard, the cell will still die and it'll break apart, and all of the everything inside the cell will kind of get spilt out into all of the other cells and might damage some of the other cells surrounding, or may cause harm to the other cells, which isn't good. Or if you have, again, in the case of extreme DNA damage, you might have mutations which lead to cancer, which again is very bad. So in these cases, so to avoid these cases, it will very cleanly and very neatly just commit suicide through this apoptosis, which like leaves nothing to be spilt out. It controls everything, makes sure everything gets deleted and managed properly, make sure that there's no mutation, anything left over. And this process is vital to prevent genetic disorders like cancer. All cases of cancer have disabled apoptosis in order to grow unregulated. That's why it's extremely common. Very much the most common gene mutation in cancer is the p53 mutation. And p53 mutation that's the gene that controls this apoptosis, one of the main genes that controls this apoptosis mechanism. So the vast majority of cancers have a mutation in this gene which disable apoptosis or find a way to circumnavigate it or go around it. Otherwise, from their DNA damage and their rapid rate of growth, they would the mechanism would be activated and they would undergo this process. In addition to cell death, even after cell death, many animals actually, or many organisms have a thing called regeneration. Some animals, which are very common, like starfish, worms, newts, other similar species, they have regenerative abilities potent enough to regrow limbs and tails from nothing, or essentially nothing. Like if a starfish loses entire one of its limbs, it can just regrow back, right? That's what I mean from nothing, if the limb is fully removed. This process is not fully understood and is still being researched heavily, but generally it involves dedifferentiation of cells in that area into stem cells and the formation of a blastema, which is a mass of pluripotent stem cells. The blastema then undergoes rapid cell proliferation and differentiation to repair and regenerate all of the lost tissue. Essentially, all the cells in that area that got damaged, they dedifferentiate into stem cells, form a clump, and then that clump undergoes massive reproduction and proliferation, leading to many, many, many stem cells. And then each of those stem cells differentiate into all of the tissues they need, all like the skin, the organs, and everything else that you need in that area. And then they just migrate to wherever they need to go. It is so far thought that blastemas can differentiate into any cell type except neurons, meaning that neural organs cannot be regenerated. So this goes back to like the whole zombie movie trope. If if you cut off a zombie's arm, it'll either regrow back or it can keep going. If you hit a zombie in the brain or you destroy its brain, it can't regenerate that and it dies. Same thing with animals. Like if you destroy the brain or any other important neural organs, it can't be regenerated, and the organism will die. Humans do have limited regenerative capability, the most impressive of which is the liver, which may regenerate fully from removal of as much as 90% of its mass. So you can get rid of 90% of your liver, and over time, it'll actually go back to its original, regenerate back to its original size. However, it is not thought to use pluripotent stem cells or blastema, but rather uses the existing mature hepatocytes that are already there, and it undergoes proliferation of those hepatocytes, which regenerate the kidney. So it doesn't form the blastema, it doesn't de-differentiate back to stem cells. It just uses the existing hepatocytes as a base to regenerate. There are other organs such as ribs, vas deferens, and in some cases fingertips, which can also be regenerated. If you break a rib, or if you remove a rib, actually, if you undergo vasectomy, sometimes the vas deferens can regenerate. Fingertips, if it's shallow enough of a cut, you can regenerate a fingertip. Induced pluripotent stem cells are a big step into regenerative medicine, and it is a very heavily researched and fast-growing field at this time. No huge technology has been released as of yet. When I say that, I obviously mean there's plenty of big research and huge research with huge implications coming out all the time in this field, in regenerative medicine. But what I mean is there's no technology where you can regrow a kidney from scratch. There's no technology where you can regrow a brain from scratch. There are things which are coming kind of close, specifically organoids, which they've done with a couple of organs. Essentially, they're clumps of stem cells which can function as or perform functions similar to an organ, like a kidney. But they're not there yet, and they can't be like used in people yet, and they're not efficient yet. And so that's what I mean with that. But this is a very fast-growing and very popular field now, regenerative medicine. Since the advent or the discovery of these induced pluripotent stem cells, which are huge for biology, one of the biggest discoveries in medicine in the past 20, 30, 40 years, along with, obviously, we talked about before, CRISPR-Cas9, which is another big thing. Okay. So that's it for the lecture for today. We will try to get the PowerPoint and recording out to you. Okay, please email me. Thank you very much for your time, and have a great night.